So I will start the presentation with a description of the principles of drug resistance. So 80 years ago, Salvador Luria and Max Delbruck published a really brilliant paper describing the frequency of resistance of bacteria to virus infections with a virus-sensitive strain of bacteria. And what they showed is that about 1 in 100 million bacteria was resistant in a population of wild-type sensitive bacteria. And by showing this, they made the important observation that resistance in a microbial population is a rare event that pre-exists in that population and is not something that is induced by exposure to the population. For this work and others, the two of them got a Nobel Prize, but what they observed applies to drug resistance, antibody resistance, and so on, as we'll discuss. So virus replication results in changes, that's mutations, allowing for the virus to adapt to the presence of selective pressure. And this is true of all life forms. There is nothing perfect about replication, whether it's viruses or humans. Errors occur because we're talking about thousands to millions of nucleotides that have to be replicated. RNA viruses have a very high mutation rate, about 1 mutation per genome in a small genome, while in higher eukaryotes like us humans and our other vertebrate animal friends, that rate has to be very low because we have very big genomes, and we don't want to have to generate hundreds or thousands of offspring to get a good one. And so there are differences in the fidelity of our replication processes. And I should point out that the rates of mutation are a function of the magnitude of replication and the fidelity of the replication. And higher eukaryotes, double-stranded DNA genomes, whether bacteria or higher animals, have a proofreading mechanism that corrects some errors and reduces the mutation rate. So this is a figure that I generated for hepatitis B virologists, but it applies to all microbes. And that is the Darwinian principles of virus evolution and drug resistance, that mutagenesis is replication dependent. So you need a reproduction of the microbe. The reproduction is associated with errors in the genome sequence, and that results in genetic diversity. With viruses, when somebody is infected by a single virus of, let's say, HIV, over time, there are mutations that accumulate in those viruses that are replicating, and the total population of genetic variants in an individual is called the quasi-species. The quasi-species is not a subset, it's the total genetic variants of that individual. With those genetic variants, in the presence of a selective pressure, like an antiviral drug, the more resistant viruses are more fit. Darwinian survival of the fittest, and they go on to reproduce in the presence of selective pressure. And this principle applies to monoclonal antibodies or immune responses, as we've seen with COVID over and over again. And as a result, HIV evolution in 1 year in one person equals all of human evolution over 1 million years, because of the higher magnitudes of reproduction and the higher error rate. So let's get to antiviral drugs. The first one was AZT, and all we had was one drug. So we conducted a study to see whether that would benefit patients, because the first drugs tried were they all had no efficacy and some had toxicity. So mortality at the end of the study, 24 weeks, one of 145 of the AZT recipients had died, and 19 of the placebo recipients, and the study was halted. And as you can see, the various subsets in the right here of subjects all showed a statistically significant benefit in terms of survival. Because my colleagues and I were interested in drug resistance based on experience with herpes drugs, we took sequential samples from patients here in San Diego and showed that over time, the virus you isolated from patients became progressively more resistant to AZT, and that was associated with the progressive accumulation of mutations in the resistant virus. And what was observed is that over time, the patients who were on AZT started to fail as well, because the drug no longer had any substantial activity against HIV. The next class of drugs that came along were the non-nucleoside RT inhibitors, and nevirapine was the first to be approved. And what we did in our lab was look at, once we knew that drug resistance readily emerged with AZT, the nucleosides, we looked at the non-nucleosides in a cell culture and showed that the potency of the drug with multiple passages just in a petri dish, the virus became progressively more and more resistant to nevirapine. And we showed that that even after one passage in the presence of drug, you could find virus with resistance. And to prove that the mutations that we saw was the mutation, it wasn't just random evolution and mutations. There was at least initially the 181 mutation in reverse transcriptase. When you put it into a wild type virus by molecular means, conferred the same resistance. So what that does is prove two things. One is that mutation accounts for the resistance, and secondly, that the target of the drug is in the reverse transcriptase. So in the phase 1 trials of nevirapine, what we observed was that the viral RNA dropped dramatically. This was actually the first demonstration of the dynamics of reduction of RNA, and within a week, one had a dramatic reduction of virus, and the CD4 went up very dramatically. But within 2 weeks, we could see the mutation emerge that we had observed in cell culture, and associated with the emergence of the drug resistance mutation, the RNA went back up to baseline and the CD4 came back down to baseline. So what we have learned is that single mutants are produced daily. They pre-exist, and one can find them in the wild type population even before the virus from an individual has seen drug. With great effort, one could actually find them as rare isolates, and then later, as deep sequencing became available, and one could screen large numbers of viruses, you could find drug resistance mutations in the original wild type population. And these are from treatment naive patients who were infected before antiviral drug was available, so we knew that it hadn't been selected for. Double mutants are far less common. The probability of 2 mutants is the product of the 2 probabilities. And then 3 or more specific mutations in the same HIV genome, often spread in different target for antivirals like the protease and the reverse transcriptase, are extremely rare. It's hard for that many mutations to appear. And this is the principle of combination therapy. This principle was first shown for tuberculosis, where any single drug selects for resistance and multiple drugs work because of the rare pre-existence of drug-resistant mutants. So knowing that drug resistance occurs, we have learned that it is the result of drug failure. The emergence of drug-resistant virus is an inevitable consequence of the failure to fully suppress HIV or multiple other microbes with antimicrobial therapy. And it's the cause of failure because once you have it, it's a major factor contributing to treatment failure. And in the mid-90s, we showed that drugs were less effective, and some of this is because people added a single drug to a failing regimen. And then Susan Little here in San Diego showed that in patients who presented within the first month or 2 of their infection, primary infection, who had not yet seen treatment, were progressively showing more and more transmitted drug resistance. And these were data accumulated in 10 North American cities. So you will hear more about how the selection for drug resistance and the transmission of drug resistance has dramatically changed over time from Dr. Toriani. From Dr. Ceccarini Silverstein. And the Merck 035 study started in the early 90s when Indinavir came along, was the first to show what you could do with combination therapy. So the standard of therapy at that time was a combination of AZT and 3TC. And in green, you can see the dramatic impact of over a log reduction within a week. But then the gradual loss of activity as the HIV RNA went back up towards baseline. Now, with AZT3TC, you can see with continued AZT3TC, one still did not get back to baseline. And this has been shown to be attributable to the impact on replication fitness of the M184V mutation of 3TC. There is the benefit to the virus to have this mutation is that it can replicate in the presence of AZT3TC, but it's not as replication fit as healthy as a wild-type virus in the absence of drug. Indinavir had a dramatic impact as single monotherapy, but over time, but a much longer time, it went back towards baseline, and that was attributable to the higher genetic barrier and the requirement for multiple mutations to develop resistance to the protease inhibitors. And then the combination of all three resulted in a reduction to below the limit of detection, which at that time was 500 copies, and that was sustained for 2 years. So if you look at the proportion of people who are undetectable, you could see that AZT3TC didn't succeed in the long run. Indinavir got about 20-25% of people. Those were people who were 100% adherent and had very low baseline RNA levels at the initiation of treatment. But you can see that with the triple combination, sustained suppression was effective. There were some failures, and people lost to follow-up were also not counted. So what we know is that as you have increasing antiviral drug activity and the selective pressure increases, that increases the probability of selecting for resistance. But at a certain point, as you get more and more antiviral drug activity, the virus replication diminishes, and with less replication, you start reducing the probability of selecting for resistance. And in fact, if you completely suppress replication, the probability of resistance is 0. It's like putting the virus in a freezer. So with the new highly recommended regimens, when 2 second-generation integrase inhibitors were compared, you can see we're getting highly successful, this is as treated, efficacy. The people who drop out of a study and so on exceed the number of people who fail. This is attributable to more potent drugs, better tolerability, and 1 pill once a day. So one other point I'd like to make is based on the Nadia trial, and Dr. Schaefer will be telling you more about this trial, which I think is a very important trial. But the only point I want to make is that people who fail AZT3TC and nevirapine, who then went on to treatment, who developed a resistance to 3TC, the question has been, what do you do with patients who are failing a regimen with these nucleosides? And the point is, if you fail with tenofovir and 3TC, going to AZT, which is not cross-resistance with the 65 mutation, really didn't make a difference. You can, the bottom line is a tenofovir 3TC or FTC regimen can be reused with a better accompanying drug, whether it's boosted darunavir or dolutegravir. And there's a reason why that is. Dr. Schaefer will discuss this study more, but the only point I wanted to make is that the 3TC mutation that results from the M184 mutation that results from 3TC or FTC hypersensitizes the virus to the activity of both AZT and tenofovir. And so you can see that there's a remarkable, more greater sensitivity of virus to, let's say, 3TC alone with no tenofovir mutations. But you even have activity with 3 or 4 mutations that are AZT associated, is close to a wild type tenofovir alone. And with 3TC 0 or 1 mutations, the 3TC resistance zidovudine still has activity. So the point I want to make here is continuing tenofovir with a 3TC resistance mutation is a good idea. And secondly, even with drug resistance, there is residual partial activity of these drugs, which is important when one is trying to compose an optimal regimen for people who have multiple drug resistance. You may not have completely new drugs, but you have to combine even partial activities to get an optimal regimen. So to conclude, mutations resulting in resistance occur as part of a viral replication and reproduction of all life forms. Selective pressures like drug treatment or immune responses select for the most fit genetic variants. Darwin was right. Viruses generate very large populations so that all variants emerge during replication. To prevent the emergence of drug resistance, potent drug combinations are needed to treat multiple resistant variants in a large population. Drug resistance is thus both a result and a cause of treatment failure. And resistance mutations confer a spectrum of diminished susceptibility. Optimal regimens for treatment failure may often need to include drugs with diminished activity.